Okay, let's unpack this. Welcome to a deep dive where we are taking on the ultimate command center, the nervous system.
Right.
And we aren't just memorizing parts here. We're going to trace its entire existence,
Right.
starting with how it's built, structure by structure, right up to the complex wiring that, controls your entire life.
It's ambitious for sure, because the nervous system is this perfect storm of what we call localization of function.
Meaning what, exactly?
It means everything you do, from consciously deciding to move your arm, which is a somatic function, to the stuff you never think about, like breathing or digestion, the autonomic functions. Every single one is mapped to a very specific spot in the brain or spinal cord.
And that's the challenge, right? Because if you just look at an adult brain, it's just this mass of gray and white matter all folded up. It seems kind of random, almost impossible to figure out why one part is next to another.
Exactly, which is why we have to use a developmental framework. It's the only way it makes sense. If we build the system from scratch, starting with that tiny embryonic tube, the logic behind all of the connections in both the central nervous system, so the brain and spinal cord,
And the peripheral nervous system.
right, everything else, it all becomes much more clear. It's like having a roadmap that shows you how everything got to where it is today.
So our mission today is basically to build the nervous system from the ground up. We're assuming zero prior knowledge. We're going to define every core term and hopefully turn a long list of definitions into a really compelling story about your own anatomy. It's just wild to me that this whole command center starts out as, what, a simple strip of tissue?
Not even. It's just a simple layer of cells. We're starting right at the beginning of life, about 16 days after fertilization, when you just have three primary embryonic layers, the germ layers.
Okay, what are they?
So you have the innermost layer, the endoderm, that's going to form the linings of your internal systems, like your gut and your respiratory tract.
And the middle one.
That's the mesoderm. Think muscle, connective tissue, most of your skeleton. It's the middle stuff.
And finally, the outer layer.
The ectoderm, and this is the kicker. The ectoderm, the outermost layer, obviously forms your skin, what we call the integumentary system, but it also forms your entire nervous system.
So our command center actually develops from the outermost layer of the embryo. That's counterintuitive.
It really is.
So how does a piece of this ectoderm turn into the brain?
Well, it's this incredible piece of anatomical engineering. A specific region of that ectoderm gets a signal and it thickens up. It differentiates into something called neuroectoderm, which forms a flat structure called the neural plate right along the back of the embryo.
Okay, so it starts as a flat plate.
Yep. Then molecular signals tell that plate to start buckling. Imagine you take a flat rug and you lift up the middle. You'd create a trough, right?
Oh, valley, yeah.
That's a neural groove. And the raised edges of that groove, the walls of the valley, those are the neural folds.
And I'm guessing those folds keep rising.
They keep rising, they move toward each other, and then they fuse over the top. It's like a zipper closing up. And in doing so, they pinch off a hollow structure right underneath the skin. That hollow structure is the neural tube.
And that tube is the key.
That is the fundamental distinction. The neural tube is the precursor to the entire central nervous system, the CNS. The front end of it balloons out to become the brain, and the long, straight back part becomes the spinal cord.
And it's hollow. Does that space stay there?
It does. Its
Yep.
hollow center becomes the ventricular system that we'll talk about later. It's where all the cerebrospinal fluid flows.
Okay, so if the tube is the CNS, the brain and spinal cord, where does the peripheral nervous system, the PNS, come from?
Great question. As those neural folds are coming together and fusing, some special cells right at the crest of the fold, they don't get incorporated into the tube. They cluster on the sides. This tissue is called the neural crest.
And what do these neural crest cells do?
They're amazing. They actually break away and migrate. They travel all throughout the developing body.
So they're not staying put.
Not at all. And the neural crest forms almost all of the peripheral nervous system. The ganglia, the nerves, all that wiring. But here's the really weird part. They don't just make nervous tissue.
What else do they make?
Some of them form the cartilage and bone of your face and skull, the craniofacial bones. Others turn into melanocytes, the cells that produce the pigment for your skin and hair.
That is bizarre. So the same block of cells that makes my peripheral nerves also makes a part of my skull and my freckles.
Pretty much, yeah.
So once that neural tube is formed, you said the front end starts to balloon out.
Right. But this happens fast. By about 3 weeks, that anterior end swells into 3 distinct sac-like bumps. We call these the primary vesicles, and their names all use the Greek root encephalon, which just means brain.
What are the three?
First, the one at the very front is the prosencephalon, or the forebrain.
Okay.
The one in the middle is the mesencephalon, the midbrain.
Makes sense.
And the one at the back is the rhombencephalon, the hindbrain.
Forebrain, midbrain, hindbrain. Simple enough.
But it doesn't stay that simple for long. By the 5th week of development, those 3 primary vesicles expand and divide into 5 secondary vesicles. The complexity just ramps up.
Okay, so how do they divide? Let's start with the forebrain.
The prosencephalon divides into 2 new structures, the telencephalon and the diencephalon.
And what do they become?
The telencephalon grows massively to become the cerebrum. That's the big folded part of the brain you always think of, the seat of higher thought.
And the diencephalon.
The diencephalon becomes the central core, structures like the thalamus and hypothalamus.
Okay, here's a really critical piece of information. The diencephalon also forms the eye cup, which later develops into the retina of your eye.
So the retina, the part of my eye that actually senses light, is technically brain tissue.
Exactly. It's a piece of the central nervous system that develops and then ends up functioning as a peripheral sensory structure. It's a really unique situation.
Okay, so that's the forebrain. What about the midbrain, the mesencephalon?
The mesencephalon is the odd one out. It's the only primary vesicle that does not divide. It just stays the midbrain, even in the adult. It's a small but really critical region.
And the hindbrain, the rhombencephalon.
That one also divides into 2, the metencephalon and the myelencephalon.
And those become...
The metencephalon gives rise to 2 major structures, the pons, and right behind it, the cerebellum, which means little brain.
And the very last one, the myelencephalon.
That just becomes the medulla oblongata, which is the part that connects the brain to the spinal cord.
So if we connect this to the adult brain, we can actually define the brainstem. It's the midbrain, the pons, and the medulla, all stacked on top of each other.
And the cerebellum, even though it comes from that same region as the pons, is this huge separate structure for coordination.
Right. And this whole expansion process gives us what we call the neuraxis. It's the central line of the nervous system from front to back. In a 4-legged animal, that would be a pretty straight line.
Not in us.
No, because we stand upright, the human neuraxis has a major kink in it, a sharp bend called the cephalic flexure. It's right between the brainstem and the forebrain.
And that's why the layout of the adult brain can seem so confusing. It's not a straight line at all.
Precisely. And we have to remember that the neural tube was hollow. That space inside doesn't go away. In the adult CNS, it remains as the ventricles.
Which are filled with fluid, right?
Yep, they're interconnected spaces filled with cerebrospinal fluid, or CSF, which acts as a shock absorber. And the shape of the ventricles mirrors the embryonic divisions perfectly.
Okay, this whole story is fascinating, but it's not just academic. It has huge clinical importance if something goes wrong, like if the neural tube doesn't close all the way.
Absolutely. That defect is called spina bifida. The neural tube is supposed to be completely fused shut by about 28 days after fertilization. If the back end fails to close, you get a spinal defect.
And there are different levels of severity.
Yes, a whole spectrum. The mildest form is spina bifida occulta. Occulta means hidden. In this case, the vertebrae don't fully close around the spinal cord, but the cord itself is fine.
So you might not even know you have it.
You might not. Sometimes the only sign is a little patch of hair or a dimple on the lower back. There are usually no neurological problems.
What's the next step up?
The more serious cases involve tissue actually protruding out. In meningocellae, the protective layers around the cord, the meninges, poke out through the hole in the bone and form a fluid-filled sac.
But the nerves themselves might be okay.
They might be. The symptoms can be less severe, but the most critical form is myelomeningocele.
And what happens there?
In that case, the meninges and the spinal nerves, or even a piece of the spinal cord itself, are inside that protruding sac. The nervous tissue is exposed and damaged.
Which would lead to really severe, permanent neurological problems below that spot.
Exactly. It requires immediate surgery to try and minimize the damage.
Okay, that was a really fast-paced look at the nervous system's blueprints.
It was. So, for a quick memory trick, if you want to remember the adult structures from the 3 primary divisions, forebrain, midbrain, hindbrain, just use the initial letters of the 5 secondary vesicles. It's TEL-D for the forebrain, MES for the midbrain, and MET-MYL for the hindbrain.
And a common confusion people have. It's really easy to think the neural tube forms all nervous tissue.
But it doesn't. Remember that split. The tube is for the central nervous system, the brain and spinal cord. The neural crest is for the peripheral nervous system ganglia and nerves, plus those weird non-nervous tissues like facial bones and melanocytes.
Okay, time for a quick check question. Which primary vesicle of the embryonic nervous system does not differentiate into more vesicles at the secondary stage?
That would be the mesencephalon, the midbrain. It's the only one that stays put while the forebrain and hindbrain each split in two.
So following that developmental path, we now arrive at the adult central nervous system. Let's start with the biggest, most dominant structure, the one that came from the telencephalon.
The cerebrum. That massive, wrinkled, gray surface, that's the foundation for all of our higher functions. Consciousness, memory, emotion, thought, it's all happening there.
And the cerebrum is split right down the middle, isn't it?
Yes, by a very deep groove called the longitudinal fissure. This creates the right and left cerebral hemispheres.
But they have to be able to talk to each other.
They do, and they use a huge bridge of white matter fibers deep inside called the corpus callosum. It's the information superhighway between the two halves.
And that wrinkled outer layer is called the cerebral cortex. You said it's wrinkled for a reason.
Yes, cortex just means bark. And it's wrinkled for one simple reason: surface area. If you were to iron out all those folds, the human cortex would cover about 1 square meter.
Wow.
The folding is just a brilliant engineering solution to pack that huge sheet of processing power into the small space of your skull.
So we have names for all these folds and grooves, right?
We do, and they're surprisingly consistent from person to person. A ridge or a convolution is called a gyrus. The plural is gyri.
And the groove in between is a sulcus.
A sulcus, chloral sulci. And we can use a few very large, very consistent sulci as landmarks to divide the brain into its 4 major lobes.
Okay, let's map it out.
All right. Find the deep groove running along the side of the brain horizontally. That's the lateral sulcus. Everything below that is the temporal lobe.
Got it.
Now find the prominent groove that runs vertically from top to bottom. That's the central sulcus. It's a huge landmark. Everything in front of it is the frontal lobe.
And everything behind it must be the parietal lobe.
Exactly. And then at the very, very back, you have the occipital lobe, which is specialized for vision.
So each lobe has a main job, more or less?
A primary mandate, yeah. Frontal is for motor control and executive function. Parietal is for body sensation, what we call somatosensation. Temporal is for hearing and memory. And occipital is purely for visual perception.
Okay, but we can get more specific than just lobes, can't we? I've heard of Brodmann's areas.
Yes, Brodmann's areas. Over a century ago, a German neuroanatomist named Corbinian Brodmann painstakingly mapped the entire cortex into 52 different regions. He did this based purely on what the cells look like under a microscope, the cytoarchitecture.
So he wasn't looking at function, just structures?
Just structure. But what's absolutely remarkable is that his structural map aligns almost perfectly with functional specialization. It's incredible.
So let's use the central sulcus as our guide again. What's happening on the sensory side behind it?
Okay, so in the parietal lobe, the ridge immediately behind the central sulcus is called the postcentral gyrus. This is Brodmann's areas 1, 2, and 3, and it's the primary somatosensory cortex.
Yeah, this is where we feel things.
Everything. All your general body sensations land here. Touch, pain, temperature, itch, but also the really important senses of proprioception, your sense of where your body is in space, and kinesthesia, your sense of movement.
Okay, what about the other sensory areas?
In the temporal lobe, you have areas 41 and 42, which are for primary auditory sensation. And in the occipital lobe, areas 17 and 18 are for primary visual perception.
Now let's jump to the other side of the central sulcus, into the frontal lobe, for motor control.
Right. The ridge immediately in front of the central sulcus is the precentral gyrus. That's area 4. This is the primary motor cortex.
And what does it do?
This is mission control for movement. It contains the cells called upper motor neurons that send the final commands down your spinal cord to make your skeletal muscles contract.
But doesn't the brain have to plan the movement first?
It does. And that happens just in front of the primary motor cortex in a region called the premotor area. This area plans the sequence of movements, figures out how to do it, and then hands the final program over to the primary motor cortex to execute.
What about language?
Also in the frontal lobe, a region called Broca's area, areas 44 and 45, is responsible for language production. It's what translates your thoughts into the specific motor commands needed for speech. And for about 95% of people, it's only on the left side of the brain.
And then there's the very front of the frontal lobe, the prefrontal part.
The prefrontal lobe. This is the highest level of cognitive function. Executive planning, your personality, short-term working memory, your consciousness. It's all rooted here. We know how critical it is from the tragic history of the prefrontal lobotomy, which severed connections here and drastically changed patients' personalities.
Okay, now let's go deeper, underneath the cortex.
Right. We have these deep clusters of gray matter called subcortical nuclei. We've mentioned the hippocampus and amygdala for memory and emotion, but for motor control, the key players are the basal nuclei.
And you have to be careful with the name here, right?
Yes, people often call them the basal ganglia, but that's technically wrong. A ganglion is a cluster of cell bodies in the peripheral nervous system. These are deep inside the CNS, so they are nuclei.
And what's their job? It's often misunderstood.
Very misunderstood. They do not start a movement. The cortex does that. Instead, the basal nuclei act like the brain's gatekeeper. They constantly compare the cortex's desire to move with the body's current state and then decide if a movement should be allowed to happen right now. They modulate the likelihood of movement.
Okay, so who are the main players in this system?
You have the C-shaped caudate, the putamen, and the globus pallidus. We often group the caudate and putamen together and call them the striatum. That's the main input station.
And this whole system is basically a circuit with two competing pathways.
Exactly. One pathway is like an accelerator and the other is a brake. But first, two simple rules. Rule 1. Input always comes from the cerebral cortex and goes only to the striatum. It's an excitatory signal.
Okay, rule 2.
Rule 2. Output always comes from part of the globus pallidus, the GPI, and it goes to the thalamus. And this output is always inhibitory. It's always trying to shut the thalamus down.
Okay, so with those rules, let's do the accelerator path first.
That's the direct pathway. The striatum fires, and it sends an inhibitory signal to that GPI output structure.
Wait, so it inhibits an inhibitor?
Exactly. And inhibiting an inhibitor is called disinhibition. It's functionally the same as excitation. So the thalamus is now freed from its normal inhibition. It can fire its own excitatory signal back up to the cortex, which ramps up motor activity. The gate is opened.
So the direct pathway promotes movement.
Correct. Now for the brake, the indirect pathway. This is the default path. It's a longer loop with more stops, but the end result is that it reinforces the GPA's normal inhibition of the thalamus. So the thalamus stays quiet, it can't excite the cortex, and motor activity is decreased. The gate stays closed.
So how does the brain choose which path to use, the accelerator or the brake?
It uses a chemical switch, dopamine. Dopamine is supplied by a small structure in the midbrain called the substantia nigra pars compacta, or SNC.
And what does dopamine do to the pathways?
It does two things at once. It sends dopamine to the striatum, where it activates the direct pathway, the accelerator, and inhibits the indirect pathway, the brake.
So when dopamine is present, you're pushing the gas and letting go of the brake at the same time.
Precisely. Movement becomes likely. When the SNC is firing, you're in an active, ready state. When it's silent, the brake path dominates and movement is suppressed.
And this is exactly what goes wrong in Parkinson's disease.
Tragically, yes. Parkinson's is caused by the death of those dopamine-producing neurons in the SNC.
So there's no dopamine.
No dopamine, which means the direct pathway can't be activated and the indirect pathway can't be shut off. The basal nuclei get stuck in brake mode. The result is a hypokinetic disorder, hypo meaning less, movement.
Which is why patients have tremors, rigidity, and so much trouble just starting to move.
Exactly. Their muscles aren't the problem. Their internal gatekeeper has the emergency brake pulled permanently. And the main treatment, LDOPA, is just a precursor to dopamine that can get into the brain to try and restore some of that balance.
Wow. The basal nuclei really are the silent decision makers behind every single action.
They are. So for a memory trick to remember the main gyri, use the central sulcus as your line in the sand. Motor is pre, as in pre-central gyrus. Sensory is post, as in post-central gyrus.
And for a common confusion, a lot of people think the basal nuclei start movement. They don't. They modulate or suppress it. If they started movement, Parkinson's patients would have too much movement, but they have the opposite problem.
Right. And for a quick check question, which cortical area located in the frontal lobe is responsible for planning a movement before the motor command is executed?
That would be the premotor area.
Okay, moving deeper into the core of the brain, past the cerebrum, we find the diencephalon. It's actually kept its embryonic name, which means through brain.
And that name makes sense because it's like a central hub, right?
It's the required connection point for almost everything passing between the cerebrum and the rest of the nervous system. You can think of it as a gatekeeper that fences off the cerebrum from just a flood of unfiltered information.
The two main structures here are the thalamus and the hypothalamus. Let's start with the thalamus.
The thalamus is this huge collection of nuclei that acts as the mandatory traffic control center for all of your senses.
All of them.
Well, almost all. Before any visual, auditory, or body sensation can reach the cortex for you to become aware of it, it must stop at the thalamus first.
And it's not just a passive relay station, is it?
Not at all. That's a key insight. The thalamus actively filters and processes all that data. It helps decide which information is important enough to bring to the cortex's attention. It's essential for focus.
You said almost all senses. What's the exception?
Olfaction, the sense of smell. This is the only sensory system that connects directly to the cerebrum without going through the thalamus first.
Why is that?
It's believed to be an evolutionary holdover. The earliest vertebrate brains were basically just advanced olfactory bulbs. Smell is probably our oldest, most primal sense, and it retains that direct line to the cortex.
Okay, and just below the thalamus is the hypothalamus.
Right, and if the thalamus is the sensory hub, the hypothalamus is the command center for homeostasis, keeping your internal body stable. It's the executive region that coordinates your autonomic nervous system and controls your entire endocrine system through its connection to the pituitary gland.
So your body temperature, your hunger, your thirst, that's all the hypothalamus.
All of it. Now, below the diencephalon, we get to the brainstem. This is the tapering cone that connects the big brain to the spinal cord. It's made of the midbrain, pons, and medulla.
And its job is basically to keep us alive.
Absolutely. It governs our most fundamental, non-negotiable survival functions. Heartbeat, breathing rate, things you never want to have to think about.
The topmost part is the midbrain.
Yep. The midbrain is divided by a little channel, the cerebral aqueduct, into a roof, the tectum, and a floor, the tegmentum.
The tectum is the interesting part, with those 4 bumps.
The 4 bumps, the colliculi. The 2 lower ones, the inferior colliculi, are just relay stations for the auditory pathway. But the 2 upper ones, the superior colliculi, are fascinating.
What do?
They integrate sensory maps. They take in visual, auditory, and somatosensory information to build a map of the space around you. Their job is to instantly orient your head and eyes toward a sudden stimulus.
So if a door slams behind me, my head whips around to look. That's my superior colliculus at work.
That is your superior colliculus. Below the midbrain is the pons. Pons is just Latin for bridge.
And it's a bridge to the cerebellum, right?
Exactly. It's defined by these massive bundles of fibers connecting to the cerebellum, which makes sense because they both come from the same embryonic vesicle. The pons is the main input route for the cerebellum.
And at the very bottom, we have the medulla oblongata.
Right, which is continuous with the spinal cord. It houses some really important nuclei. One is the reticular formation, which is a diffuse network that regulates your sleep-wake cycles and your general level of alertness.
Okay, now let's talk about that little brain attached to the back, the cerebellum.
The cerebellum. It's only about 10% of the brain's mass, but it contains over half of the brain's neurons. Its sole job is motor coordination. It makes sure your movements are smooth, precise, and corrected on the fly.
So how does it do that? You called it a real-time motion GPS.
That's a good way to think of it. It's constantly running a comparison loop. It's checking if what you intended to do matches what your body is actually doing.
So it needs 2 pieces of information at the same time.
Exactly. It needs the plan and it needs the reality. First, the plan. When your cerebrum sends a motor command down to your spinal cord, say, to start walking, it sends a complete copy of that plan through the pons to the cerebellum.
So the cerebellum knows the intention.
Right. Second, the reality. At the very same moment, all the sensory feedback from your body proprioception from your joints, balance information from your inner ear, is rushing back up. It goes through a nucleus in the medulla called the inferior olive and then into the cerebellum.
And the cerebellum compares the 2.
It compares the plan to the reality. If you step on an uneven bit of pavement, the feedback immediately shows a mismatch. The cerebellum instantly calculates the needed correction and sends a new command via the brainstem to the spinal cord to adjust your muscles.
And this is all happening completely unconsciously.
Instantly and unconsciously. It's a silent correction system. And this explains why there's no direct connection between the cerebrum and the cerebellum. All the information has to be routed through those specific brainstem pathways.
Okay, this section covered the critical relay and correction centers.
For our memory trick, think about the midbrain colliculi. The superior colliculus is higher up and handles the more complex job of integrating sight and sound for orienting. The inferior colliculus is lower down and just handles the simpler auditory function.
Common confusion. Viewing the thalamus is just a simple switchboard.
Right. It's not passive at all. It's actively filtering, prioritizing, and processing information. It's the reason you can focus on one thing in a noisy room.
And a quick check question. Which structure of the diencephalon is the only sensory pathway that bypasses the thalamus and connects directly to the cerebrum?
That would be the olfactory system, your sense of smell.
All right, let's move down the neuraxis to the final major piece of the CNS, spinal cord.
The spinal cord. Unlike the brain, which got so complex, the spinal cord really keeps the basic structure of that original neural tube. It's a long cylinder, basically an information highway with two-way traffic.
On the outside, it has a couple of key landmarks.
It does. There's a deep groove on the front or anterior side, that's the anterior median fissure, and a shallower one on the back, the posterior median sulcus.
And this front-back split is really important functionally, isn't it?
It's fundamental. It goes all the way back to the embryo. The back, or dorsal side, developed from the alar plate, and it is purely for sensory functions. The front, or ventral side, came from the basal plate, and it is all about motor functions.
And you can see that where the nerves attach.
Perfectly. Sensory information always enters the spinal cord through the dorsal, posterior nerve root. Motor commands always emerge from the spinal cord through the ventral anterior nerve root.
Here's a critical point. The spinal cord doesn't run the whole length of your spine.
No, it doesn't. Your bony vertebral column keeps growing through childhood, but the spinal cord itself stops growing when you're about 1 or 2 years old. It ends up terminating way up in your lower back, around the L1 or L2 vertebra.
So what's below that?
The spinal nerves still have to exit at their proper level, so below the end of the cord, they just hang down in a big bundle before they exit. This bundle of nerve roots looks like a horse's tail, so it's called the cauda equina.
And this is why a lumbar puncture or spinal tap is safe.
Exactly. They insert the needle way down low, below L3, where there's no solid spinal cord to damage. They're just pushing aside the nerve roots in the cauda equina to get a sample of the cerebrospinal fluid.
Okay, now if we slice the spinal cord and look at a cross-section,
you'll see the gray matter is in the center, in a butterfly or H shape. This is where the cell bodies and synapses are. The processing happens here. And we call these regions the gray horns.
what are the different horns?
The posterior, or dorsal horn, is the receiving area for all incoming sensory information. The anterior, or ventral horn, is the motor command center. This is where you find the cell bodies of the big motor neurons that send signals out to your skeletal muscles.
And there's a third one sometimes.
Yes, the lateral horn. It's only found in the thoracic and upper lumbar regions. It contains the cell bodies for the autonomic nervous system motor neurons, the ones controlling your involuntary functions.
And the white matter surrounds all of this.
Right. The white matter is organized into white columns. This is all myelinated axons, bundled together into tracks for long-distance communication up and down the cord.
So you have tracks going up and tracks going down.
Yep. Ascending tracks carry sensory information up to the brain. They're mostly in the posterior columns. Descending tracks carry motor commands down from the brain. They're found in the anterior and lateral columns.
Okay, time for a quick review of that vital information highway.
The memory trick for this is a classic. Sensory dorsal, motor ventral. Think of it this way. You feel things with your back. Dorsal posterior is for sensory. You move the big muscles on the front of your body. Ventral anterior is for motor.
And the common confusion is assuming the spinal cord runs the whole length of the spine.
Right. Always remember, it stops high up, around L1 or L2, leaving the cauda equina hanging below. That's what makes a lumbar puncture safe.
All right, our quick check question. Which spinal cord horn contains the cell bodies of motor neurons that regulate involuntary functions like heart rate and digestion?
That would be the lateral horn.
Here's where it gets really interesting. The central nervous system is so vital, so priceless, that the body has built multiple layers of incredible protection around it.
Specialized protection. You have the bony skull and vertebrae, of course, but then you have these tough membranes, a liquid cushion, and a very privileged blood supply.
Let's start with the blood supply. The brain is greedy for oxygen.
Incredibly greedy. So the arterial supply is redundant. It comes from two major pairs of vessels. First, you have the internal carotid arteries, which come up through the neck and enter the skull.
And second,
the vertebral arteries. And these are really protected. They actually travel up through little holes in your cervical vertebrae before entering the skull at the foramen magnum. Once inside, they merge to form one big basilar artery.
and these two systems meet up at the base of the brain.
They do. They form a unique structure called the circle of Willis.
And this is a safety feature.
It's an essential safety feature. It's an arterial circle that ensures that even if one of the main arteries gets narrowed or blocked, blood from the other side can still get to all parts of the brain. It maintains constant perfusion.
And when that system fails, that's when you have a stroke.
That's a stroke. a disruption of blood supply, usually from a clot, that causes brain tissue to starve and die. And the effects are so specific depending on where it happens. It's why we use the FAST mnemonic: face drooping, arm weakness, speech difficulty, time to call for help.
So after the brain uses the blood, how does it drain out?
The venous blood drains into these really big channels called dural sinuses. They're basically large veins that are enclosed within the protective layers of the brain covering.
And they all eventually funnel the blood out of the head.
Yep. They drain into the big jugular veins in your neck to return the blood to your heart.
Okay, let's talk about those protective coverings themselves. The meninges.
Right. The brain and spinal cord are wrapped in 3 layers of these connective tissue membranes.
Layer 1, the outermost.
That's the dura mater, which means tough mother. And it is. It's a thick, fibrous, leather-like layer that's anchored to the inside of the skull. It's so tough it even forms partitions that keep the brain from sloshing around too much.
And it encloses those dural sinuses we just talked about.
Exactly. The middle layer is the arachnoid mater. It's named that because it has these delicate spiderweb-like filaments, the arachnoid trabeculae, that stretch down from it.
And the innermost layer.
That's the pia mater, the tender mother. It's a very thin, almost transparent membrane that is shrink-wrapped directly onto the surface of the brain. It follows every single gyrus and sulcus.
And the space between the arachnoid and the pia is really important.
That is the subarachnoid space. It's vital because this is where the cerebrospinal fluid circulates. This is the liquid cushion.
And if these layers get inflamed, that's meningitis.
Yes, usually from a bacterial or viral infection. And it's so dangerous because the inflammation causes pressure on the whole CNS, leading to headache, neck stiffness, and it can even damage nerves coming out of the brain.
So that brings us to the cerebrospinal fluid system itself.
The CSF system, this is the other major layer of protection. It cushions the brain, but it also acts as a kind of lymphatic system, clearing out metabolic waste.
Where does this fluid come from?
It's produced in all 4 ventricles by a special structure called the choroid plexus. This is a membrane full of specialized glial cells, called ependymal cells, that filter the blood to make CSF. You make about half a liter of it every day.
And it flows in a one-way path through the ventricles.
A very precise path. It goes from the 2 big lateral ventricles in the cerebrum down into the 3rd ventricle in the diencephalon, through the narrow cerebral aqueduct in the midbrain, and into the 4th ventricle at the level of the brainstem.
Then what? How does it get outside to cushion the brain?
From the 4th ventricle, there are 3 little holes or apertures that let the CSF exit the ventricular system and flow out into that subarachnoid space. There, it bathes the entire outer surface of the brain and spinal cord.
So it's made inside, then flows outside. How does it get drained?
It filters back into the blood. There are these little outpouchings of the arachnoid mater called arachnoid granulations that poke up into the dural sinuses. They act like one-way valves, letting the CSF drain back into the venous blood flow.
And this whole system is different from the blood-brain barrier.
Completely different. The blood-brain barrier is a chemical barrier that protects the brain tissue itself. The CSF system is a mechanical cushion and a waste removal system.
Okay, let's review the CNS protection infrastructure.
Our memory trick for the main arteries is to think of their initials. I for internal carotid and V for vertebral. Together they supply the brain, IV.
And the common confusion is mixing up the blood-brain barrier and the CSF system.
Right. Remember, the blood-brain barrier provides chemical privilege. The CSF system provides mechanical cushioning and waste removal. 2 different but equally vital jobs.
Time for the quick check question. Which specialized structure in the ventricles, composed of ependymal cells, is responsible for filtering blood to produce CSF?
That would be the choroid plexus.
All right, we've built the CNS and protected it. Now let's wire up to the rest of the body with the peripheral nervous system.
We finally leave the CNS. The PNS is everything outside the brain and spinal cord. It's all the wiring. And it's made up of 2 main things: bundles of axons, which we call nerves, and clusters of cell bodies, which we call ganglia.
Let's start with ganglia. What are they?
A ganglion is just a collection of neuron cell bodies out in the periphery. We can categorize them by function. For example, sensory ganglia contain the cell bodies of sensory neurons.
Like the dorsal root ganglion.
The dorsal root ganglion is the classic example. It's that little bulge on the dorsal nerve root right before it enters the spinal cord. It holds the cell bodies of all the sensory neurons, bringing information in from your body.
And then there are autonomic ganglia.
Right. These contain the motor neuron cell bodies that control all your involuntary homeostatic functions.
And there's a special system in the gut, right?
Ah, yes, the enteric nervous system, or ENS. It's this incredibly complex network of ganglia and nerves embedded right in the wall of your digestive tract. It's often called the second brain because it can manage most of digestion on its own without much input from the CNS.
Okay, so that's ganglia. What about nerves?
A nerve is just a bundle of axons in the PNS, and they are protected by layers of connective tissue, kind of like a telephone cable.
What are the layers?
The outermost tough layer around the whole nerve is the epineurium. Inside, the axons are bundled into little groups called fascicles.
And each fascicle has its own wrapping.
Yep, the perineurium. And then finally, each individual axon inside a fascicle is surrounded by a delicate layer called the endoneurium.
Okay, now for the big players in the PNS, the 12 cranial nerves.
The 12 pairs of cranial nerves, these connect directly to the brain, mostly the brainstem, and they handle almost all the sensory and motor functions of your head and neck.
There's a mnemonic to remember their names, right?
There is. The classic one is, on old Olympus's towering tops, a Finn and German viewed some hops. That gives you olfactory, optic, oculomotor, and so on.
And there's another one for their function, whether they're sensory, motor, or both.
Yep. Some say marry money, but my brother says brains, beauty matter more. That helps you keep track of which are which.
So let's categorize them.
Okay, there are 3 that are purely sensory. Olfactory for smell, optic for vision, and vestibulocochlear for hearing and balance.
And how many are purely motor?
There are 5 that are purely motor. 3 of them control your eye movements, the oculomotor, trochlear, and abducens. Then you have the spinal accessory for your neck muscles and the hypoglossal for your tongue.
And that leaves 4 that are mixed. They do both sensory and motor.
4 mixed nerves. The trigeminal for face sensation and chewing, the facial for facial expressions and taste, the glossopharyngeal for your throat and taste, and then the big one, the vagus nerve.
The vagus nerve, CNX. This one is the exception to the rule.
It's the superstar anomaly. Vagus means wandering, and it's the only cranial nerve that leaves the head and neck. It wanders down into your chest and abdomen to control the autonomic functions of your heart, lungs, and digestive tract. It's the main highway of the gut-brain axis.
Okay, so those are the cranial nerves. What about the nerves coming off the spinal cord?
The spinal nerves, these are much more regular. We have 31 pairs of them.
And unlike the cranial nerves, they're all the same type.
Every single one of them is a mixed nerve. They all contain both sensory axons coming in the dorsal root and motor axons going out the ventral root.
But once they leave the spine, they do something interesting. They reorganize.
They do. In most regions, the axons from different spinal nerves weave together to form these complex networks called nerve plexuses. A plexus is just a network of fibers, no cell bodies, that allows for reorganization. From the plexus, new systemic nerves emerge to supply the limbs.
And there are 4 major plexuses.
4 main ones. The cervical plexus in the neck, which gives rise to the phrenic nerve that controls your diaphragm.
Then the big one for the arms.
The brachial plexus, an incredibly complex network that gives rise to all the major nerves of the arm, like the radial, ulnar, and median nerves.
And then the lower body.
You have the lumbar plexus for the anterior leg, which gives us the femoral nerve, and the sacral plexus for the posterior leg, which forms the massive sciatic nerve.
Is there any place that doesn't form a plexus?
Yes, the thoracic region. The spinal nerves T2 through T11 don't form a plexus. They just emerge directly as the intercostal nerves and run between your ribs.
Okay, that covers all the wiring of the peripheral nervous system.
It does. So for a memory trick on cranial nerve function, sensory motor, or both, remember, some say marry money, but my brother says brains, beauty matter more.
And a common confusion. Don't mix up a ganglion and a plexus.
Right. A ganglion is a cluster of cell bodies. A plexus is a network of nerve fibers, or axons, bodies versus wires.
And finally, our quick check question. Which large network of peripheral nerve fibers gives rise to the radial, ulnar, and median nerves?
That is the brachial plexus.
Now let's test our foundational knowledge by running through a rapid review of the core concepts we covered today, citing the source material for accuracy. First up, question 13. Aside from the nervous system, which other organ system develops out of the ectoderm? The correct answer is C, integumentary.
That's right, C, integumentary. The ectoderm is the outer germ layer, and it gives rise to both the nervous system and the skin. The other options come from the endoderm or mesoderm.
Question 14. Which primary vesicle of the embryonic nervous system does not differentiate into more vesicles at the secondary stage? Answer is B, mesencephalon.
Correct. B, mesencephalon. The mesencephalon or midbrain is the only one that stays undivided. The forebrain and hindbrain both split in two.
Question 15. Which adult structure arises from the diencephalon? A, thalamus, hypothalamus, retina.
Yep, it's A. The diencephalon is the secondary vesicle that forms the thalamus and hypothalamus, and as we discussed, the retina is a unique extension of this same CNS tissue.
Question 16. Which non-nervous tissue develops from the neuroectoderm? D, craniofacial bone.
Correct. D, craniofacial bone. This is the work of those migrating neural crest cells, which come from the neuroectoderm and form things like facial bone and melanocytes.
And question 17. Which structure is associated with the embryologic development of the peripheral nervous system? A, neural crest.
A, neural crest, absolutely. The neural crest is what breaks away to form the PNS. The neural tube, option D, forms the CNS.
All right, next set. Question 18. Which lobe of the cerebral cortex is responsible for generating motor commands? That's D, frontal.
D, frontal, for sure. The frontal lobe is home to the primary motor cortex and the premotor area, which plan and execute movement. The other lobes are sensory.
Question 19. What region of the diencephalon coordinates homeostasis? C, hypothalamus.
C, hypothalamus. That's its main job, regulating the autonomic and endocrine systems to keep the body stable. The thalamus is the sensory relay.
Question 20. What level of the brainstem is the major input to the cerebellum?
B, pons. B, pons. The pons is the bridge to the cerebellum, relaying that copy of the motor command from the cerebrum so the cerebellum can do its comparison.
Question 21. What region of the spinal cord contains motor neurons that direct the movement of skeletal muscles? A, anterior horn.
That's A, anterior horn, also called the ventral horn. It's the gray matter region with the cell bodies of the motor neurons that control your voluntary muscles.
And question 22. Brodmann's areas map different regions of the two particular functions. B, cerebral cortex.
It's B, cerebral cortex. Brodmann's map is specifically about the microscopic structure of the surface gray matter of the cerebrum.
Okay, group 3. Question 23. What blood vessel enters the cranium to supply the brain with fresh, oxygenated blood? C, internal carotid artery.
Yes, C, internal carotid artery. It's one of the two main pairs of vessels, along with the vertebral arteries, that feed the circle of Willis.
Question 24. Which layer of the meninges surrounds and supports the sinuses that form the route through which blood drains from the CNS? A, dura mater.
A, dura mater. The dura is the tough outer layer that physically encloses and supports those dural sinuses where the venous blood collects.
Question 25. What type of glial cell is responsible for filtering blood to produce CSF at the choroid plexus? A, ependymal cell.
A, ependymal cell. Ependymal cells are the specialized cells in the choroid plexus that line the capillaries and produce the cerebrospinal fluid.
Question 26. Which portion of the ventricular system is found within the diencephalon?
B, third ventricle.
E, third ventricle. The third ventricle is the space right in the middle of the diencephalon, between the two halves of the thalamus and hypothalamus.
And question 27. What condition causes a stroke? D, disruption of blood to the brain. It's D, disruption of blood to the brain. A stroke is, by definition, an interruption of blood supply, which leads to tissue death from a lack of oxygen.
Last group. Question 28. What type of ganglion contains neurons that control homeostatic mechanisms of the body? C, autonomic ganglion.
C, autonomic ganglion. Autonomic ganglia, like the sympathetic chain ganglia, are where the cell bodies for the involuntary homeostatic control system are located.
Question 29. Which ganglion is responsible for cutaneous sensations of the face? D, trigeminal ganglion.
D, trigeminal ganglion. The trigeminal nerve, CNV, handles facial sensation, and its sensory cell bodies are located in this ganglion.
Question 30. What is the name for a bundle of axons within a nerve? A, fascicle.
A, fascicle. Axons in a peripheral nerve are bundled into fascicles, which are then wrapped by the perineurium. A tract is the CNS equivalent.
Finally, question 31. Which cranial nerve does not control functions in the head and neck? D, vagus.
D, vagus. The vagus nerve, CNX, is the wandering nerve. It's the one that goes down into the chest and abdomen to control the organs there.
We've completed the full anatomical story of the nervous system.
We really have. We tracked it all the way from a simple ectodermal strip up to the final arrangement of 31 spinal nerves and 12 cranial nerves. We've mapped the cognitive centers, the gatekeepers of movement, and the brain's incredible protective systems.
It's a system defined by its capacity for flawless, integrated action.
Exactly. And we focused a lot on the cerebellum's role as that silent, unconscious proofreader the real-time motion GPS.
Right. It demands a copy of your conscious intention from the pawns and instantly checks it against reality from the inferior olive.
To make corrections before you even realize you've made a mistake.
So what does this all mean?
Well, think about what happens when the cerebellum is damaged or when that timing loop breaks down. You get ataxia, uncoordinated, jerky, stumbling movements. It tells us that this silent system isn't about thinking about moving. It's about perfecting the action of moving. The fact that our feeling of being coordinated is a function of a completely unconscious deep brain comparison loop suggests that our experience of smooth, intentional movement is, in a way, an illusion. It's an illusion built on a foundation of constant, hidden, instantaneous correction. It is a system built not for thinking, but for constantly perfecting the doing.